All right, everybody, should we get started? Yeah. Excellent. Well, in beginning this presentation, I want to welcome you all to North Carolina Museum of Natural Sciences. My name is Chris, and I'm the coordinator for current science programs here at the museum. And I get the pleasure and privilege of welcoming all of you to Darwin Day. Isn't that fantastic? I hope that you've had a great day exploring the museum, taking in all of the games, crafts, exhibitors, and vendors that have come out to the museum today, especially for today's very special theme, fungus. Everybody likes a fungus, right? Maybe some of them. Maybe some of them you don't like too much. And all day long, we've also been hearing from guest experts who've been delivering fantastic presentations. And I think for this round would be no different. Now, this presentation is going out two places. It's being broadcast live onto the internet right now. So I can wave hi to all of the internet audience who's tuned in. But that does mean that when we get to the Q&A portion of the event after the presentation, if you want to ask a question and you're in the room taking the presentation in person here on site at the museum, you'll be on the live stream. Not necessarily your face, but at least your voice and your question will go out onto the internet. If you would like to ask a question and not have that happen, you can just let me know and I can ask your question and get that into the live stream as well. I'll also be watching the chat on YouTube. So for our visitors watching on the live stream, you can leave your thoughts, comments, questions, and experiences as well. I'll be monitoring that to pass those questions along to today's guest speaker, who I think at this point, I should introduce. Those in the room and watching online, you can see him on the screen now. Dr. Bryn Dentinger is with us from the Natural History Museum of Utah and the School of Biological Sciences, which I hear is the second best museum in the United States. You can decipher for yourself what the best natural history museum in the world is. Yes, it's the one that I work for. But I'm excited for today's presentation and curious of where a psilocybin-themed presentation is going to take us. So, Dr. Dentinger, I'll turn it over to you.
All right, thank you very much for that introduction. I hope everybody can hear me all right. Can I get a thumbs up from you if that's all good? Cool. So first of all, let me just say we poached Jason Cryan from your museum to become the director of ours. Jason says hello, and I come in peace. So the title of my talk, Adventures with Psilocybin-Producing Mushrooms, New Insights on Their Evolution and Diversity, is a thinly veiled reference to a children's book, which is depicted by the image here in the lower right corner. This comes from Alice in Wonderland, Lewis Carroll's Alice in Wonderland, or at the time when he sketched this out, it was called Lewis Carroll's Alice's Adventures Underground. And I grabbed a couple of these original drawings that Lewis made that you'll see throughout the talk. Now, Alice in Wonderland is a great reference to talk about Adventures with Psilocybin-Producing Mushrooms, because there's this famous scene in the book where Alice partakes in a mushroom and it alters her perception of reality. And that's precisely what psilocybin-producing mushrooms do. So before I launch into that part of the talk, I was curious when I was asked to deliver this talk what the significance of the date of November 12th was for Darwin. And it turns out there wasn't any significance. It was just a matter of convenience. But I thought it would be fun to think about where Darwin was on November 12th during his voyage to the Beagle. And in 1832, he was in Montevideo, Uruguay. And here's a quote from one of his notebooks, one of the captain's logs that he wrote in on the 12th of November. I think Montevideo looks different today than it did when Darwin was there. He was also back there in 1833. So both November 12th, 1832 and 1833, Darwin was in Uruguay in Montevideo. He wasn't there for the entire year, of course. He was traveling around, but he had returned. In 1834, November 12th, he was off the coast of Chile and could see the eruption of the Osorno volcano, which is pictured here. And in 1835, he was on a boat again. He spent a long time on a boat over those 5 years in French Polynesia, around these islands, and wrote to Tahiti. So this is where Darwin was on November 12th. I wouldn't mind being in this place today, but one interesting thing about Darwin that I also wanted to mention is that unlike a lot of his peers at the time, he actually collected fungi during his voyage on the Beagle. Most of the natural scientists that were out collecting in that era didn't pay any attention to fungi. Fungi at the time were classified as lower, quote unquote, lower plants, and they really weren't given much attention, certainly not the attention they deserve. And so I thought it's significant that Darwin actually did pay a little bit of attention to them and brought these specimens back. The specimens pictured on the left here are all deposited at the University of Cambridge in the UK. And this picture on the right is my favorite collection that he made. This is his handwriting on the note. This is a fungus that became known as Cyteria darwinii, named after him, of course, which he collected in Tierra del Fuego, which is the southernmost part of Patagonia. And this fungus is a very interesting fungus. And in fact, he was so fascinated by it that he wrote about it in his voyage of the Beagle. And he wrote a whole paragraph on this fungus, and that's quite unusual. And here's a nice drawing that was included in the voyage of the Beagle. In fact, I was so enamored of it that I got it tattooed on my arm. Okay, but as far as we know, bringing it back to the topic that I was asked to speak about today, as far as we know, Darwin did not collect any psilocybe or psilocybin-producing mushrooms. But that's just as far as we know. We only have the records that he wrote down, so who knows what he was doing over those 5 years on the boat, really. Okay, so I wanted to give you a bit of a timeline on the human psilocybin symbiosis. So there's some evidence, although it's still quite questionable, that humans had interactions with psilocybin-producing mushrooms many thousands of years ago. And this is indicated by some rock art that is found in southern Algeria. And it depicts what some people have interpreted as shamans with mushrooms sprouting from their bodies, which they've interpreted as an indication that people were consuming psychedelic mushrooms. But really, the only real first written evidence we have of the use of psychoactive mushrooms, psilocybin-producing mushrooms, that is, comes from the 16th century. And this was recorded in these codexes that were brought back by some of the first Spanish explorers of the Americas. And in particular, this Spanish friar, Sahagún, wrote about them and was able to identify these mushrooms as, in the local language at the time, called teonanacal, which I'm sure I'm butchering the pronunciation, which translates roughly into God's flesh. So this was the name that the indigenous peoples of Mexico gave to these magic mushrooms, so-called magic mushrooms. It wasn't until the early 20th century, however, that scientists really picked up the ball. So there were hundreds of years of basically this knowledge being lost to Western science. And it wasn't until anthropologist Reiko and ethnobotanist Schultes kind of picked up the pieces from the early 16th century texts and identified the teonanacal mushrooms as Paniolus, in the genus Paniolus. But it turns out that they were both wrong at the identity. And it wasn't until 1951 when Rolf Singer, a famous mycologist who visited the Farlow herbarium at Harvard, where Schultes and Reiko had deposited their specimens, re-examined them and re-identified correctly this time the samples as Psilocybe cubensis. However, this information was still largely relegated to the scientific literature, and it was not popularly known about the existence of these psychedelic mushrooms. That changed in 1957 with R. Gordon Wasson, who was an ex-banker who became fascinated with the uses of mushrooms around the world. He learned from Schultes about the existence of the ritualistic use of psychedelic mushrooms in southern Mexico, and so led an expedition down there in the 1950s and documented it and he also partook in the ritual, as you can see here depicted in the photograph of him accepting psychedelic mushrooms from a shaman in southern Mexico. And R. Gordon Wasson published this in a popular article in Life magazine in 1957, and that's when he coined the term magic mushroom. At this time also, the famous chemist Albert Hofmann, who was responsible for discovering LSD, isolated and determined the structure of the compound psilocybin. And this is the active compound in psychedelic mushrooms that causes the effects in humans when they are consumed. This brought in what was known as the psychedelic era of the 60s and 70s. So the popularization of this led to a lot of people experimenting with psychedelic mushrooms during this period. However, this came to a sudden end in 1971 when Richard Nixon declared a war on drugs, enlisted psilocybin and many other substances on the Schedule I as Schedule I of the Controlled Substances Act. And this meant that there was no approved medical use for these chemicals, and they were therefore illegal to possess. This really suppressed research on these drugs then since 1971. And so then we entered what I call the era of ignorance, where we just pretend they don't exist and we choose not to learn anything about them. And this is largely due to this war on drugs from Nixon's presidency. However, today we're having a sea change in society's perception and view of these kinds of drugs. In fact, it's becoming clear that psilocybin is proving to be one of the most effective antidepressants and other therapeutic agent for a wide range of other mental illnesses, and it may be an incredible tool for psychotherapy in the future. And so this is really changing the way that society views these chemicals from being these evil drugs that can ruin society to drugs that in fact may be hugely beneficial, especially now when we have this massive mental illness crisis on our hands. And this has led to more popular accounts of the uses of psychedelics, like this book by Michael Pollan that came out a few years ago. And it's also led to a number of ideas and discussions about how important this might be to mental health in the US. And here's another example of an article that was published covering this topic on the heels of Oregon passing a law that legalized psilocybin in the state. In fact, there's been a trend lately where psilocybin has been decriminalized or even legalized, even as recently as this last election we had last week. Colorado passed a bill to allow psilocybin to become legalized in the state. And this trend has resulted in psilocybin mushrooms being available for purchase online, even delivering. Here's a couple of examples of that from a company in Vancouver, where you can go and buy pumpkin spice mushroom tea or coconut cranberry mushroom chocolates, if that's more your flavor. And in the description, you can see that in the tea, it comes with 0.5 grams of golden teachers in each tea bag. And in the chocolates, it's 1.1 grams of golden teacher mushrooms. And this really led me to ask the question, which is sort of the motivation for this research. And that is, what even are golden teachers? And what does consuming half a gram or 1 gram of them really mean? We're entering this phase in society where there's a lot of interest, a lot of anecdotes and hearsay, but I think very little scientific information. So golden teachers, if you don't know, are a strain of Psilocybe cubensis. This is a species of psychedelic mushrooms. And like Alice, who ate one side of a mushroom and grew shorter and ate the other side of the mushroom and grew smaller, they can transform your perception of reality if you consume them. But like Alice, which side is which? And of course, there is no side to a mushroom because it's round, and that's part of the point here. But I think what I'm trying to communicate is that we really need to understand these organisms much better than we currently do. So what are they? So Psilocybe mushrooms, this is a genus of mushrooms. They're found worldwide. They are the largest group of mushrooms that naturally produce this compound, psilocybin. They're mostly what we call pileate stipitate lamellate LBMs. Now, what that means is they have a cap, that's the pileus, they have a stalk, that's the stipe, and they have gills underneath. And those are called lamellae, those are the scientific terms. LBM refers to little brown mushroom. And this is a catch-all term that people use for little brown mushrooms that are all very difficult to distinguish from one another. Now, one feature they have that is makes them distinguishable is that they have purple-brown spore prints. That's it. That is, if you take a cap and you put it on a piece of paper and cover it with a cup and let it sit overnight, when you come back in the morning, if you take the cap off, you will have spores deposited on that paper. And the color of that deposit can be a useful character for identifying mushrooms. And in this case, it has a purple-brown color that is distinct for this group of mushrooms. There's also one weird form known as psychoactive, and that's depicted down here. Hopefully you can see my arrow. It's the photo of the mushroom in the lowermost left. And that is a mushroom that never opens up, so you don't get gills, and the spores are not dispersed by the wind. Instead, the spores have to be dispersed by other means, and we think it's likely that this is being dispersed by animals, but we don't know that yet. Now, what are they doing in nature? Well, they're all saprotrophic, meaning they decompose dead organic material, and this is plant material in this case. And so they're found in soil, in wood, and on dung. Many of them are also associated with disturbed areas, and therefore they may be pioneer species. There's about 300 globally distributed species that have traditionally been classified in the genus Psilocybe. 165 of those have this characteristic bluing reaction. That is, when you pick them and handle them, they turn blue. And I'll be talking more about that later on. And it's thought that that blue color indicates the presence of psilocybin, the psychoactive drug. The center of diversity is in the temperate and tropical zones of the Americas. And that might have to do with where the mycologists live that study them, but it may also be a real pattern. And of course, they have a long history of cultural use in Mesoamerica, as illustrated by these mushroom stones from Guatemala in the lower left-hand corner. In 2002, there was a landmark study of the mushroom-forming fungi, where this team produced the largest phylogeny, that is an evolutionary tree of the mushroom-forming fungi to date. And what they found was that those 300 some odd species of Psilocybe, the ones that they sampled, sorted into two groups that were not each other's closest relatives. And they noticed that one group was composed of all the species that have that bluing reaction, and the other group was composed of the species that didn't have the bluing reaction. This led them to propose the name Psilocybe for the group that has the bluing color, because of the long history of use of this name associated with the psychedelic species. And the other species then were reclassified into the genus Diconica. However, that reclassification remains unfinished, and only 37 species of the remaining 150 or so have been transferred. So there's still many species that haven't been transferred yet. Now you can see that the morphology of these is quite similar in appearance. Even at the microscopic level, you can get spores like these here that are the same shape roughly as spores as these in this group. And that is just another illustration of which there are many in the fungi, where morphology just is not adequate for classification purposes. Now, I have a confession to make, which is that I've only encountered Psilocybe mushrooms 9 times in my career. That's not very many. These are the 9 encounters I've had since 2002 with Psilocybe. Now, you might be wondering at this point, what makes me an expert to talk to you about these mushrooms today? And the truth is, it's not my own personal expertise. It's the expertise of the people that work with me. So especially this person, Dr. Alexander Bradshaw, it's his work that I am presenting today. He's done all of the hard work for this. We are also heavily dependent on the taxonomic expertise and knowledge of these 2 collaborators in Mexico, Dr. Laura Guzman and Dr. Virginia Ramirez. Laura Guzman is the daughter of the famous mycologist Gaston Guzman, who is the most knowledgeable person on Psilocybe mushrooms. He passed away, sadly, but she's taken up his role and is providing us with all of the important expertise we need to be able to do this project. 2 other people are essential to the success of our project as well. Juliana Furci helps us with gathering specimens and helps us with translating our science into something more palatable for the public. And Paul Stamets, that visionary mycologist, has been a core part of our team and has provided many important resources to allow us to do this research. But it's not just the people that are important to us, it's also the institutions. Institutions like mine here at the Natural History Museum of Utah, at the North Carolina Museum of Natural Sciences. And that's because museums are the strongholds of biodiversity. They're the custodians of these physical vouchers that allow us to do these projects. So they are dedicated, accessible locations where these specimens can be stored for long-term purposes. They're rich, so these collections are rich sources of metadata, so it tells us where something occurred at a particular time. And this kind of information can be really important as baselines for understanding how things change over time, say in response to climate change and other phenomena. These storage places, these rooms, are climate controlled, and they're protected to protect the specimens for long-term future study. And it's because of this that we are able to do the research that I'm presenting on today. So I've divided the talk into 3 parts. The first part, I'm going to tell you about authenticating Psilocybe in fungarium specimens, as well as examining how well their chemistry is preserved. I'm then going to shift and talk to you about phylogenomics, which I know this is a mouthful, but this is really understanding the evolutionary history, the family tree of Psilocybe, which then allows us to understand something about how psilocybin evolved. And then I'm going to switch gears entirely away from my empirical data, and I'm going to start to speculate with very little evidence about the functional role of psilocybin in nature. Okay, part 1. So we first had to find and locate species that we wanted to work on. And luckily, this all happened when all of our labs got shut down. And so Alex, Dr. Alexander Bradshaw, spent his time during the lockdown on his computer collating information from online public repositories. And so what he did is he was able to find specimens of Psilocybe in 72 different institutions around the world. That totaled 3,690 specimens that were collected over the last 139 years, or at an average rate of 26 samples a year. We requested every single one of them and received about 1,600 of them, so a little less than half of what we requested we received for study. This represents 61 years of collections. Now, if you remember from a few slides ago, I showed you my encounter with Psilocybe over the last 20 years. And over 20 years, I encountered it 9 times. It would take me more than my career's worth to be able to gather samples of this quantity to be able to do the kind of research we need to do. And so that is why these institutions, the museums, the herbaria, that hold these specimens are so important. Okay, so what we did is we set out to figure out whether the specimens were correctly identified or not. And to do this, we sequenced a portion of the genome that's known as the barcode region. And this in fungi is the internal transcribed spacer, or ITS. It's just a small fragment of the genome that varies enough that it's unique to a species in most cases. So what I've done is I've put these into a phylogenetic tree to see where the barcodes, how the barcodes are related to other sequences of mushrooms. And the red color here indicates when a barcode sequence was placed in a genus other than Psilocybe. So this would be specimens that were identified as Psilocybe that turned out to be in a completely different genus. The yellow color indicates when a species was correctly identified as Psilocybe, but it was incorrectly identified as at the species level. And the blue indicates where the species of Psilocybe were correctly identified. So in total, we found that misidentification was rampant throughout the specimens. 13% of them, in fact, belong to 5 different genera other than Psilocybe. And most of the correctly identified specimens belong to only 3 well-known species of Psilocybe, that being Psilocybe cubensis, Psilocybe cyanescens, and Psilocybe semilanceata. And those are indicated here with the letters. So you can see these are all Psilocybe cubensis, these are all Psilocybe cyanescens, and these are all Psilocybe semilanceata. So there's very few blue, so correctly identified to species that weren't one of these 3. We also wanted to know whether the psychoactive compounds and their derivatives were stable in these collections. So the motivation for this really was that there were a number of institutions, when we asked for loans of these specimens, they refused to send them, citing the legal status of psilocybin. And so we were curious, does the compound really persist in these old collections? And nobody had ever asked that question before. So we partnered with a natural product chemist here, pictured on the right, Dr. Jackie Winter, and she helped us develop an assay using GCMS profiling with chemical standards so we could accurately detect and quantify these different metabolites in the mushrooms. And we chose 3 species to focus on. The first here that I'm showing you is Psilocybe cubensis. And we had specimens that ranged in age from around 15 to 50 years old. And the y-axis is showing you the concentration of the different metabolites that are pictured at the top. And you can see that these metabolites were virtually undetectable in all of the specimens, except for a specimen that was 42 years old. There was a very small amount of psilocybin detectable in that specimen. This strongly suggests that these metabolites do not persist in old fungarium collections, at least after 15 years of storage. However, the same is not true when you look at this species, Psilocybe cyanescens. Here you can see that there's a lot of variation from specimen to specimen, which is not correlated very well with time, with age. So we have a specimen that was very young, a year or 2 old, where we see large concentrations of psilocybin in the specimen. But a specimen that's only 8 years old has virtually no detectable levels of psilocybin, just very little psilocybin present. So this is indicating that there's just so much variation from specimen to specimen that you can't really predict, based on the identity of the specimen or the age of the specimen, whether or not it will have these compounds. Finally, we see similarly in Psilocybe semilanceata that these compounds can be found in older preserved specimens, but in this case, the specimens that were 40 plus years old had no detectable levels. And it's unclear if that is a time-dependent trend or if this is just, again, some of the vagaries of the variation found in specimens from specimen to specimen. Curiously, though, in the younger specimens, up to 20 years old in this species, the psilocybin content seems to be more stable than and more predictable than in the other two species. We don't know why that is, but it might have to do with initial preservation or condition of the material before it was preserved. Okay, so the take home from this part is that the fungarium specimens labeled as psilocybe are frequently misidentified, even to the rank of genus. And so they can't be used, the names can't be used as predictors of the presence of metabolites, whether they're controlled metabolites or not. And furthermore, psilocybe and the related metabolites are so variable from specimen to specimen that you can't predict, even knowing the species, if it has it in it. And in some cases, maybe not at all. And these results, although they're preliminary, they really have important implications for how these specimens are stored, shared, cared for, and used by researchers, and as well for whole organism recreation and therapy. That is, in states like Oregon and Colorado, which are now facing legalization of this compound and the potential sale and consumption of whole mushrooms, the way that the mushrooms are preserved could impact the therapeutic and recreational value of them. Okay, so I'm going to shift now to part 2. But before I get into the nitty-gritty, I need to explain some things. So I'm going to give you a little bit of background here. The first is how psilocybin is synthesized in the mushrooms. So the reason that psilocybin is psychoactive is because its structure resembles very much the structure of serotonin in our brains, and therefore it's able to interact with serotonin receptors in our brains. In fact, it's not psilocybin that's doing that. It's this compound here, psilocin. This is the active metabolite. So psilocybin is called a prodrug. When it's consumed, it gets dephosphorylated. So this P and O gets removed, leaving only an O and H, and that's this compound psilocin. This is what actually gets absorbed in the brain and interacts with the serotonin receptors. It turns out that this biosynthesis of psilocybin in the mushroom wasn't known until 2017. And it was finally elucidated by this group, and they showed that psilocybin is the result of an enzymatic conversion from the amino acid tryptophan using 4 enzymes. So there's one enzyme that encodes a decarboxylase, and that's labeled Psi-D, and that's the first step. There's a second enzyme that encodes, a second gene that encodes an enzyme that's a hydroxylase that performs the second step. A third enzyme that encodes a kinase that phosphorylates it. That's the third step. And then there's a fourth enzyme that is a methyltransferase. So this adds methyl groups, that's this here, twice. And the end result then is you get this structure here, psilocybin. Now, a cool thing about fungi is that these enzymes, the genes that encode them tend to be clustered in the genome, meaning they're physically associated with one another. They're close together. That means that one can look at a genome and look for genes that encode enzymes with catalytic functions that are close together, and from that infer that that is the biosynthetic gene cluster. And in fact, that is the case here with psilocybin. This is the biosynthetic gene cluster in the genome of Psilocybe cubensis. And these are the genes, the kinase that performs that third step, the hydroxylase that performs the second step, the methyltransferase that performs the fourth step, and the decarboxylase that performs the first step. And they are all very close to one another in the genome. Okay, now, interestingly, it is known that psilocybin occurs not only in the genus Psilocybe, but in other species of mushrooms that are distantly related from Psilocybe. And this is an evolutionary tree that shows that. So we know that psilocybin is concentrated up here in Psilocybe, but it's also found in the genus Gymnopolis, in some species of Gymnopolis. It's found in some species of Inocybe. is found in some species of Paniolus and some species of Pluteus. And you can see that these all have different ancestors. Now, this is a pattern that would be consistent with evolutionary convergence. That is, these species may have evolved the ability to produce psilocybin independently of each other. Another study showed, just after the publication showing the biosynthetic pathway, that in fact, in two of these other distantly related species, Gymnopolis, Paniolus, and Conosibe, that the cluster is the same across all of them. So here's the gene cluster, and you can see that the same enzymes are found in roughly the same order in all of those species. And when you build an evolutionary tree out of one of the enzymes, it turns out that the enzymes show patterns of relationships that are independent of the species relationships. And this is evidence for horizontal gene transfer. What that means is it appears that these species have acquired the biosynthetic gene cluster, these genes, from another organism rather than having inherited it from their own ancestor. We were curious, though, because there were two other species that weren't included in that study, one of which is the most phylogenetically divergent species, way down here, Pluteus salicinus, and another species has a very different ecology from the rest, Inocybe corydalina. And we were curious, what about what's happening in these species? So we sequenced the genomes and then looked for the gene cluster, the psilocybin biosynthetic gene cluster. And as a reminder to you, this is what that gene cluster looks like in all of the other species identified to date. So first, in Pluteus, we found the same gene cluster, and you can see that the order of the genes is the same. However, in Inocybe, we did find the same gene cluster. Instead, we found a different gene cluster where the order of the genes that look like they perform the same functions is very different. And in fact, when we build a gene tree, the evolutionary history of the genes, this is for one of the genes, you can see that the gene from the Inocybe cluster is really different from the same genes found in all of the other clusters. This is evidence that that psilocybin biosynthesis evolved independently through convergent evolution in Inocybe corydalina. So here we have two cases where psilocybin has evolved both multiple times independently and transferred horizontally multiple times. Okay, so the take-homes from this background here are that we now understand the genetic basis of psilocybin biosynthesis. The most distantly related mushrooms, psilocybin-producing mushrooms, have acquired the gene cluster through horizontal transfer. And in one case, it appears that there's a mushroom that has evolved psilocybin biosynthesis independently. Okay, so we were curious to know, how has psilocybin biosynthesis evolved in the genus Psilocybe? As you may recall from earlier on in the talk, there's 165 species of Psilocybe that all have this bluing reaction that all presumably manufacture psilocybin. Yet we only know about psilocybin biosynthesis in 3 species. And in fact, these 3 species are found only in one part of the tree known as clade 2. So what's going on up here? What about these other species? Well, we identified where the gaps were. And one gap is that the most comprehensive phylogeny available for Psilocybe is based only on three gene regions. And that means that the relationships are not fully supported. So there's some ambiguity how things are related to one another. And that makes it difficult, impossible, in fact, for us to know for sure if we are observing the patterns, the real patterns over evolutionary time. Also, only 22 of the 144 species, or 165, depending on who you talk to, were included. So the vast majority of species still were not represented, and we don't know where they belong in the phylogeny. And importantly, also, only 3 of these species have sequences that are derived from type specimens. So I will explain what a type is in a second. So what we proposed to do was sequence whole genomes from every known species of psychedelic psilocybe and use type specimens whenever we could. So we set out to acquire all the types, and some institutions required that we visit them. And so here's Alex holding the type specimen of Psilocybe cyanescens at the Royal Botanic Gardens, Kew. Now, one problem is that type specimens tend to be old and they tend to be in poor condition. And that means they yield low quantities of poor quality DNA. That DNA is often heavily damaged. So you can see from this graph that the average size of DNA fragment is around 50 base pairs long. That's a very tiny fragment, and that makes it very hard to work with. Another problem that we face with these old DNAs is that one of the nucleotides, so if you recall, DNA is made up of 4 nucleotides, adenine, guanine, cytosine, and thymine, and one of those, cytosine, can undergo spontaneous conversion to uracil, which is the version of thymine used in RNA. Now, what happens is because of the spontaneous conversion, when we go back to read the DNA, we misread some of those cytosines as thymines. And so we get this bias indicated here by the uptick in the red line of thymine residues. And this can mislead us in downstream analyses unless we correct for this problem. Finally, the other problem we face with these old degraded materials is that very little of the DNA we get out of them is actually from the target organism. And that's because over time, these specimens can acquire molds and bacteria that end up having much more DNA in them, much more intact DNA in them than the organism of interest. And so taken together, this creates a really difficult problem for us to sort through. However, we have developed methods in my lab where we can get usable DNA sequences out of some of these old specimens. And here's an example. This is the holotype collection of Stropharia cubensis, which is now known as Psilocybe cubensis. It turns out that there was no DNA sequence available from the type. So everything that people called Psilocybe cubensis today is we can't actually tie it to the type material. So we used some techniques, and this is a chart showing you how the DNA that we pulled out of the specimen can be classified. And what I want to highlight here is we have a couple of big clouds of red and blue, and red is animal DNA and blue is bacterial DNA. The orange is the only fungal DNA we had in the sample. Now, if we look at the genus level, we can see what might have happened here a little bit. All of that blue that was red in the past one, which was animal DNA, we can now see is in the genus Homo. That's you and me. That's human DNA. So you can see the problem we face. We wanted mushroom DNA, but the majority of the DNA we get is from humans. So we have to go through a process of cleaning this. We have to filter it out and select for only the fungal DNA. And we can do that. And when we do that, we can then extract the fungal DNA and build trees, evolutionary trees from the fungal-only DNA. So we did that for 71 samples. This represented 67 species, and they turn out to be both in psilocybe and out of psilocybe. So 52 of these belonged to psilocybe, what we call sensu stricto. This is what we now currently accept as being psilocybe, the bluing species, the ones that should produce psilocybin. 16 of those landed outside in the genus Dentinger. That's that other genus that many of the other species of psilocybe are now being transferred to. And then we found 3 other species of psilocybe that didn't belong in Dentinger, but also didn't belong in psilocybe. And we don't know what genus they really refer to at this point. 20 of these genomes come from type specimens. that allows us to anchor these names for the first time. And importantly, because we used whole genomes, we were able to build a tree from nearly 3,000 genes. That gives us a lot of data and a lot of confidence in the results. And so unlike that 3-gene tree, this gene is now fully resolved.
Bryn, I'm just going to give you about a 3-minute warning.
Okay, thanks. Okay, so we also found that the origin of psilocybe was dated to 65 million years ago, and they diversified at 53 million years ago. So we were able to expand the current phylogeny of Psilocybe, and we had a couple of changes from the original tree. So we're updating this. And then we looked at the gene cluster across the psilocybin biosynthetic gene cluster. And as predicted in that clade 2, where we had genomes from 3 species, this is the order of the cluster. But when we look at the other group, we can see that the order of the genes is different. You see the colors indicate the order. And so we found that, in fact, there was a big difference in the evolution of psilocybin biosynthesis in this group. So the take home here is that the clade 1 of psilocybe has different order, and that Psi-H, one of the genes, was duplicated many times. Okay, so I'm running out of time, I guess. I may have misunderstood how much time I had to give this presentation. So I'm going to zip through this quickly because I think it's rather interesting. Basically, people think that insects might be the selective force for psilocybin. But in fact, we can find insect DNA in psychedelic mushrooms, and this is an example of that. We've also reared adult flies out of psychedelic mushrooms. Now, this points to the fact that psilocybin, if it is a defense against insects, is not very effective. Instead, I have a different idea. I think it may be that psilocybin is involved in defense against slugs and snails. In fact, take a look at this. This slug is absolutely devouring these mushrooms, decimating them. Now, that would be a very strong selective force. And so, interestingly, the slugs and snails first colonized land and diversified around the same time that psilocybin evolved. But of course, this is a correlation, not causation. But there's other lines of evidence. One is that mushrooms have inducible chemical defenses to slugs. So this is a compound that all mushrooms make. It's actually used in the food industry because it's what we recognize as mushroom odor. And slugs don't like it. They stop feeding. And this compound is produced when the cells become damaged. Well, psilocybe has an inducible reaction. It's that bluing reaction I've been talking about. And it turns out that the blue color is the result of enzymatic conversion of psilocybin into a compound that can produce reactive oxygen species in the guts and burn holes in the guts of the slugs and snails. So also, in the cases of horizontal gene transfer and convergent evolution, the enzymes that convert the psilocybin into the blue pigment also came with the cluster or evolved independently. And that suggests that it's really the blue color that matters, not the psilocybin. So my hypothesis is that it's the blue pigment that is the functional compound, not psilocybin, and that psilocybin is just a happy accident. So sorry I had to wrap that up really quickly. Hopefully there's still time for some questions, and thank you for your attention.
All right, I hope I got the audio turned back on in time for you to hear the thunderous round of applause.
You're muted, so I can't hear anything.
How about that? And I'm trying to see, make sure the live stream is fine too. The availability of specimens to study, you said it was limited, although they're now legal in a couple of states.
Correct. Yeah, so it was really up to the different institutions on whether or not they were comfortable sending us material. So there's the legal status is really unclear. It's really ambiguous on whether or not there's jurisdiction over these specimens, in part because it's unknown whether or not they contain these compounds. So some institutions took a very conservative view and said, if this is labeled as psilocybe, we're going to assume that there's psilocybin, a controlled substance in them, and they're not loan them out for study. Did that answer the question?
No, I think my understanding was the question was, since fresh mushrooms are so available, why not use those instead of ones that you're trying to obtain from that have core DNA samples?
Yeah, okay. So yeah, the question was about if fresh mushrooms are readily available, why not use those? Well, they're not readily available. They're only readily available for a few species. And of course, then we really get into legal trouble if we don't have proper licensing. So we're focused on diversity. These are species that are mostly not available. They're not cultivated by anybody. They may be represented by only one or a few collections in the world. And so that's where we were focused on. So we have to rely on these sometimes old and degraded specimens.
I completely forgot the question.
No problem. No problem. Go ahead.
How far are we from addressing psilocybin as a depression movement?
I caught some of that.
How far along are we in using psilocybin to treat depression?
Yeah, well, I'm not in the medical field, so I'm not super up to date on the current status of that, but I do know that there are approved clinical trials occurring now, and there's a number of other trials happening around the country, outside of the US as well. So I would guess, I mean, purely speculative, I would guess in 10 years there will be some sort of formal therapeutic application of psilocybin.
I read a paper from 3 weeks ago.
That's what.
That was the phase 1 clinical trial. Yeah, but I'm also not a medical researcher, so it's beyond me to break down exactly what they found beyond the headlines that you would have read. All right, I'm going to bring the last one in from the chat, which comes from Michael. Do you think there are some fungi species that have compounds that can be used to treat people with other issues like anemia?
I mean, the potential is always out there. I think the thing that we have really underexplored the chemical diversity of fungi in general, and I think mushrooms especially. The dogma is that mushrooms are not very chemically diverse, but that's clearly not the case. So I think there's many potentially medically important chemicals that are produced by fungi that we don't know about yet, and that could be used for treating all kinds of things like sickle cell anemia.
And that'll do it for today. I have to flip the room in like 10 seconds. Can we do it? Go for it.
When you were showing the change in metabolites over 50 years, does that pattern also show the phylogeny as well?
You'll have to come down here. Just so you can hear.
Can you hear me?
Yes.
So when you were showing the diagrams over the 50 years of the change in metabolites, does that pattern of the changes also showcase in the phylogenies as well?
Oh, you're talking about like the degradation or stability of the metabolites over time?
Yeah.
We haven't looked at that. So it's possible. Yeah. So it's possible that there's a lot of variation from mushroom to mushroom that's genetically or even environmentally controlled. And we haven't been able to explore that yet. So we were just scratching the surface here to see if there was any. We expected there would be a little bit at least, but there was way more variation than we anticipated. And so that's something that I think will probably become important in the future when people are trying to develop whole organism therapies, because you're going to want to know if these things vary a lot.
Excellent. Thank you. Thanks, everybody, for coming out today.
All right. Thank you, everybody.
Thanks for tuning in online as well, everybody. We'll see you at the museum sometime soon or here at the next live stream presentation. Just follow naturalscientist.org. Check out the calendar events to see what's coming up next. Dr. Dentinger, thank you so much for being with us.
Thank you very much.
Bye, everybody.